So thanks to for the invitation. Oleg invited me to make a talk, and I will tell you some very interesting story about microbial life in highly chaotropic environments, which you can find on Earth. And they are actually, okay, this is location of my institute, as you can see here. And we are just in the central, geographically, in the center of Mediterranean. And since 2002, we are going to study this area here. It's the south of Crete and west-south of Crete and south of Greece. And this place is absolutely peculiar because you can find there the lakes, hypersaline lakes, which are absolutely well formed and well conserved on the bottom of the Mediterranean Sea, and the shallowest one, which is Mikhail, located on 3 kilometers deep sea bottom. All of other, they are much more deeper, and for example, Atlanta discovery Urania at 3.7 kilometers deep. And all of them, it's hypersaline, anoxic environments. And Urania, for example, has also this part of the sea of the horseshoe. It will be northwest part, active mud volcano on the bottom. It's really absolutely incredible ecosystems. And they look like very interesting. And for example, this is one of the photo of I think it's Urania Basin, and you can see here, for example, is the beach, and here the lake is started, and all of them, they are characterized by very interesting hydrochemical parameters, because if you go inside of this lake, you see the absolutely strong increasement of salinity up to saturation, and then you have depletion of the oxygen, all of them they are anoxic, hypersaline, and enriched by sulfur. Here I will try to reproduce how they look like if you go with the ROV. This is the lake, this is discovery, which is, I will speak further about this lake, which is absolutely chaotropic with the 5 molar of magnesium chloride saturated brine. And it's actually dead. But interface is alive. And here we have this, it's courtesy of Virginia Edge Pump, where they were sampling this lake using the ROV or JSON. And you cannot imagine this, something like 13 operators with joystick. They're moving this ROV, which landed in the lake on the by the way, here, as you can see, maybe just confirm me, you can see the skull of the dolphin here. And it's really amazing ecosystem. Due to the high concentration of the salt, and that's why high density, they are separated from the seawater. As now, for example, you can see here, this is the interface between seawater and lake. And the density is so different between seawater and the brine, then the liquids, they are not mixing. For example, you can see here the manipulator entering in the lake, and then the interface will be again like reformed. It's really amazing structures. Okay, we go further. Actually, the interesting question of these lakes is their origin, because they are made very few of them, and some of them it's Gulf of Mexico, and many of them in Mediterranean Sea, and some of them also in the Red Sea, which is actually very close. If I go back, you can see here all the lakes known in Mediterranean, and actually this is really surprising because you can say that surface of Moon or Mars, it's more or better studied than the deep sea, because I was actually expertise in discover the three lakes, new lakes, Tethys, Kryos, and Hephaestus. These lakes I found, and I was first to sample biological sampling of Medea, and the size of, for example, these lakes, it's over 70 kilometers long. They are over 3 or 4 kilometers wide and up to 400 meters, for example, Medea deep. But for example, Urania, this lake is 700 meters deep after 3,700 meter where the surface is. It's really amazing systems. And actually, there are many scenarios how they were created. And one of the most feasible, I would say, it's due to large or long, more than, here you can see more than 250,000 years. It was cyclic evaporite deposition in Mediterranean Sea when the Gibraltar was close to the Atlantic Sea, and the whole Mediterranean basin became as a lake and dried off. Sometimes basin wide, sometimes with the marginal basins, and it was cyclically occurred. And in this case, in some places in the Mediterranean sea bottom or seabed, you can find the level of evaporated salts more than 3 kilometers thick. It's really amazing what was actually created. And the origin of lakes became from these evaporites, because all of them, they're located here. In this slide, I show some with the arrows, white arrows. This is the movement of the tectonical platform of the Eurasian platform and African platform. And actually all these lakes, they occur in the tectonically active zone. And now to actually situation which called evaporative path of seawater. If you will dry off seawater 10 times, the brine will have this proportion. And this is actually called thalassohaline proportion. But if you will continue to evaporate this 100 milliliters and will reach 5 milliliters of the brine became athalassohalitic or thalassohaline. And in this case, all the halite and kieserite, carnallite will precipitate, and you will have the highly enriched brine with magnesium chloride. And actually, this is the source for the lakes. This one, as I showed you before, these three lakes are filled by magnesium chloride, 5 molar magnesium chloride. Can you imagine? And absolutely huge formation, hydrological formations, both 3 of them. Okay, this is evaporative path again to explain how actually seawater can reach the hydrochemistry of, in this case, just Discovery Basin, but they are very similar: Discovery, Kryos, and Hephaestus. And all of them, they are actually fitting very well to Discovery, evaporation path of seawater, because if you are continue evaporating and using bromium as a conservative ion, you can find actually all these hydrochemical relations of the major ions, which are corresponding well to what you can find in the lakes. And yeah, again, this kind of scenario, because you have this thick evaporative layer or thick layer of evaporative salts, and some of them, like this atalasohaline, this 5 milliliters of the water which I show you, which are not absolutely precipitating, they became, or they stay in the liquid form over these 5 million years, and then due to crack, tectonical crack, they became or they go in the contact with the sediments, trying to diffuse, and this is first scenario. And second, if you have, for example, these thalassohaline lakes, if you are cracking the evaporative salts, not the brine filled with magnesium chloride. And okay, this is our most salted and most thalassohaline environments on Earth. And here we can add, it's just our old paper, and here we can add also Hephaestus Lake, which is also some kind of in between discovering Kryos deep sea lakes filled with magnesium chloride. And also I will speak very shortly about Don Juan Pond in Antarctica, which is filled with calcium chloride, about 6 molar of calcium. And it's also absolutely peculiar environment. As you can see, the salinity, it's over almost 500 grams per liter, or even in Don Juan, it can reach 670 grams per liter. And in this case, yeah, you have to speak about the gram per liter, not gram per kilo, because you have the density, as I told you. It's very, very elevated density. And for example, for ROV, which are entering in the hypersaline lakes, it's really a challenging question to enter in such a highly dense hydrological formation. But nevertheless, for example, here we have all this data relating to discovery. It's our first paper. And actually, you can see then up to 260 gram per liter of total salinity. Let me say it's here. It's in this red point. You have the really nicely formed, intact microbial cells. And we are facing here window of life of activity water, which is 0.61. But in our previous, sorry, this is about Kryos. In our first paper in about discovery lake, we found an chaotropicity window. It's corresponding to 0.79 water activity index, just because in discovery, as you can see, there is very few of sodium, much less than in Kryos. And sodium is not called chaotropic, but cosmotropic cation. And actually here you have some condensing effect or helping effect for the microbial life to survive. But in any case, below this level, we didn't find any alive microorganisms. And actually we confirmed that life is finishing somewhere here on the level around 2.6 molar of magnesium chloride. When we start, oh, there is some change in the color of the title. Okay. In any case, when we start to study the microbial diversity in these lakes, and especially with the like Hephaestus or Kryos, yeah, there are very, very fascinating uncovered world. And actually, I missed to say you this nice phrase I found from Simon Conway Morris. He said, if you want to understand aliens, stay at home. And actually, we start to study these microorganisms we published some of the interesting papers, and this is one of our first paper in Nature. And as you can see, we found, I would say this was first one, not updated at the moment, but you can see up to 12 new candidate divisions, and we called them Mediterranean Sea brine lakes, MSBL. And we reached 12 only in one paper, and then it was already about 14. But now they start to modify the taxonomy, and all of them, they already affiliated with the new type of microorganisms. And actually, what I have to say you, this is just the summary of all the lakes. But if you will take the atalasohaline or highly chaotropic lakes, then there are very few microorganisms, which we actually we're confirming all of them by RNA and not by ribosomal RNA, because ribosomal RNA in highly chaotropic environments, they are also stable. This means if you can find the pH2 some RNA, ribosomal RNA, it doesn't mean anything. There is no absolutely confirmation of active life if you cannot find the messenger RNA. And all of these red lines, they are messenger RNAs found in the level of the close to the chaotropicity window. And I would place your attention about this group, KB1, MSBL1, and halobacteria. And halobacteria, we were lucky to isolate microorganisms from the lake. This was from the Kryos, this from Medea, and both of them, they belong to this new group of absolutely previously unknown Haloarchaea, because Haloarchaea was traditionally attributed to the obligate aerobic organisms. We found obligate anaerobic Haloarchaea, and in these compartments. And some of them were isolated from the lake of in Russian Kulunda, Altai region, and I would say central Siberia. But some of them we isolated also from highly chaotropic interface of Kryos and from the Talasohaline Lake Medea. And all of them actually were very similar by 16S and by genome sequencing also. Then we just look for the database, and actually this is the bias, typical bias of the sampling, because for sure they're also here in the south hemisphere, but there is no data available. In any case, we found them in all of the regions, hypersaline, anoxic sediments, or in our case, in the hypersaline or brine lakes, anoxic brine lakes in the Mediterranean Sea. And the interesting stuff or interesting feature of them, they are sulfur respiring. This means they are using sulfur and reducing sulfur to the H2S. And actually, all of the hypersaline lakes, which we found up to now, they are sulfidic or eurohaline. And actually, this is mainly due to activity of sulfidogenic organisms. Then we try to make the modeling how actually it could happen, the ecosystem functioning in these hypersaline lakes, especially in the interface again, because down to the interface there is no life due to extremely chaotropicity and extremely low water activity of the highly chaotropic lakes, Discovery, Kryos, and Hephaestus. And actually we found some kind of model how to place together this group of organisms, halobacteria, MSBL1, and KB1. It was there not only, not from our group, but it was some confirmation about the microbial activity or metabolic activity of KB1 and MSBL1 was confirmed by other groups, including also Andreas Teske. And then we found actually very interesting stuff which we are working right now. Then if you will see this, for example, already new NGS data using non-specific primers to amplify V3, V5 regions. And actually finally we found very interesting organisms which called nanohalarchaea. And nanohalarchaea is the recently discovered new phylum from the superphylum of Halarchaea, which is from the archaea, which is absolutely a part of all other archaeal organisms, and they called the pan because they called by Diapterotritis, Altiarchaea, Diapterotritis paroarchaeota, Altiarchaea, and then Nanoarchaea, Nanohalo. It's the pan, and it's absolutely new superphylum, which actually has, I would say, less than 10 representatives in laboratory. 9 of them I have, because we are able to cultivate nanohalarchaea now in our laboratory. Okay. Surprisingly, they were very, very well correlated with the presence of the halobacteriaceae or halobacteria. And it came to the idea that maybe nanohalarchaea, because they have very small cells, they are less than 200 nanometers in diameter. They have genome less than 1 mega base pairs. They do not have basic metabolic pathways active. They can only suck from some host the nutrients. They cannot synthesize nucleotides. It's really some kind of parasitic form of life just above viruses. And then it was big accident in Italy with our ship, and actually we lost the possibility to go to these anoxic lakes and to sample them, because you have to have very sophisticated instrumentation and a big ship to sample interface with the 10 centimeters resolution, which is located more than almost 4 kilometers deep than your ship is. And it's really, tough was to do, but actually we succeed somehow. But then after accident, we lost this ship, and actually I switched to the normal salted saltern or natural salt lakes to find nanohalarchaea, and searching for the polysaccharidolytic halarchaea in some colonies. We found this very strange, by microscopy you can see very strange stuff, like it's halarchaea but someone sitting on, and actually it was exactly this nanohalarchaea, and we were really lucky to cultivate polysaccharidolytic halarchaea with the or as the host of nanohalarchaea. And here's the very beautiful images of nanohalarchaea sitting on the host. This is halarchaea. In this case, it's halomicrobium, which is chitinolithic or degrading and growing on chitin. And this is halarchaea sitting very close. Even you can see they make some deformation of the external membrane of the host cell. And I have plenty of this nice photo here. You can see even better how they can go really almost inside in the host cell. Interesting was for us then some of this Halarchaea was very isolated from the superficial lakes and the solar salter. They were very closely related to the groups of nanohalarchaea, which we found in Hephaestus deep sea lake. This means they are genetically very, very well confirmed. And yeah, they were really very, very fascinating. Then we can, without having additional access to the deep sea lakes, to study, nevertheless, to study this nanohalarchaea. Then, as we published as a new class of prokaryotes, and it was accepted in Berger manual as a candidate at nanohalobium. And actually, what we found interesting, then this teeny microorganism or teeny prokaryote. Oh, I have some. Okay, yeah. Yeah, we deciphered the metabolism. I will not go in details. Just very interesting stuff. Then, in some case, this small ectosymbiont can help host eating another polysaccharide, because, for example, this host cannot eat normal starch, but nanohalarchaea can, and that's why nanohalarchaea in some situation can help host to survive when the, for example, chitin is not present at the in this environment. And this is some kind of already symbiosis, which we published here as a symbiosis between nanohalarchaea and halarchaea based on utilization of different polysaccharides. The same we found when we started to study xylene degradation. And as you can see, as you maybe remind, all of them, they came very similar to nanohalarchaea of the highly chaotropic Hephaestus lake. This means those or these organisms, they can have the same metabolism as actually we have now in laboratory, and they were isolated from the normal solar saltern or from the superficial hypersaline environments. Also, again, with the xylene, we gain this. We again decipher how they can grow, and in this case, it was already not the binary culture, but we have some kind of three-member consortium which can degrade xylene, and then we have the couple, which is the nanohalococcus with halopherex, and they are actually scavenging the oligosaccharides from the xylene degrading halarchaea in this hypersaline and chaotropic environments. And now go to the last point, Don Juan Pond. Actually, not too many or too much to show you, because we just sampled recently I even cannot say that it's ecosystem, because actually it's dead, completely dead, but during some snow events, which are really, really rare in this place, because we're here, and it's dry valley. It's different dry valleys, which is the most driest environment on Earth, more dry than Atacama Desert, and also the coldest one, because here the average of the temperature, yeah, as Sergei Bulatman can say, in Antarctica, you are below 20 at least. In the Vostok, it's minus 55, but we are on the coast. And in this case, there is only during the winter time, you can reach minus 60. But here, it's not snow. This is not absolutely snow. It's Antarcticite. It's the crystallized calcium chloride, in form, and you can see here the crystallic hydrate, and It's really the lake is covered by the salt, by the calcium chloride. Here much more movies. Oh no, movies not. Okay. Here we were sampling. Here is the lake look like. And here you can finally see the liquid. And this is liquid we collected, and it's exactly corresponding to this salinity. This brine is 670 grams per liter of calcium chloride. And you cannot freeze it at the temperature less than minus 55. It's remain liquid. And okay, we tried to find the DNA RNA. We isolate the DNA RNA from here, and but not mRNA. Ribosomal RNA, yes. mRNA, no. And again, we considered, confirmed that all the lake here as itself is dead. But sometimes you can have the very, very strong wind from this valley, which bring the snow on this slope. And this slope then start to melt. And it's created some kind of small springs, which became immediately inhabited by microorganisms, including also the algae and I would say even the multicellular organisms you can find there. And actually, these samples we also have, but the salinity there did not increase 200. Yeah, I would say it's less than 2 molar of calcium chloride when you can see the macroscopically evidence of life. But in any case, yeah, now it's under study, and I think we will publish all this stuff. And I will start with this movie just to show you and open to questions, because the movie is 4 minutes. You can see how the interface looks like when you are going there with the buoyant ROV, which is from the Chaos University. We don't have such elevated amount of money to create such kind of equipment to go on or to fish the interface of the basins. And here is the depths of the Red Sea. One of them is Shaban Deep, brine lake, and you can see here the interface, which is highly inhabited by zooplankton as well, because all of this stuff. Here the buoyancy camera is activated, and you will see now how this row is moving just on the interface. Exactly. Here the interface. Okay, I open to the question.